Hey Saturday, this is Mr. Dubik, and in this YouTube lesson, we're going to be learning about meiosis. Meiosis is the process in which gametes, also known as sex cells, are formed. Okay, so if you look here at the diagram, or this picture, you can see that there are two different types of sex cells. There are sperm cells, and then there are egg cells. Males are going to be producing sperm cells, and females will be producing egg cells. Now in meiosis, the important part that you really need to understand is that when we go ahead and we produce and make these sex cells, the important part is that they are all genetically different. And that is the process in which we are going to talk about today, is how these gametes are formed and how they are all created genetically different. So if you look here at this picture, this is a picture of my family, okay? You can see that here to the left is me. Then if you go to the right, that's my brother, my sister, and my mom. And what you notice is that I am genetically different from my brother, my sister, and my mom. And that is the whole purpose and the point of meiosis, is to produce genetically different offspring that are all different. And the reason why we want to produce genetically different offspring is so that we're not all the same. Because if we were all the same, then we would be all susceptible to the same diseases. So it's really important that we're producing genetically different sperm and egg cells so that we can produce genetically different organisms that are all different and unique in their own way. So before we get into talking about the actual process and the different stages of meiosis, what we need to do is we need to talk about the basics and the fundamental vocabulary terms that are needed in this process of meiosis to fully understand. Okay, so what we're going to talk about first are the two different types of cells in a living organism. Okay, so we have haploid cells and diploid cells. Haploid cells are cells that contain one copy of every chromosome in a cell. Okay, so a great example of haploid cells would be sperm and egg cells. And these are these cells that we are going to be primarily talking about and focusing on in this video, because meiosis is literally the production of sex cells. And those sex cells are going to be called haploid cells because they only have one copy of every chromosome in their cells. Okay, so if you look here at my picture, you see a sperm and you see an egg cell. Okay, and each of these sperm and egg cells contain a total of 23 chromosomes. Now when they go ahead and fertilize, which they're doing, then what they're going to produce is they're going to produce a zygote, which is the first cell of a living organism, and then it's going to become a diploid cell. Okay, which contains two copies of every chromosome in a cell. Okay, so for this video, we're going to be focusing on haploid cells, and what you need to know is that those haploid cells only contain 23 chromosomes. And where do we get those 23? Is you get them from having one copy for every chromosome in all of your cells. Okay, so if you look down here in this picture right here, okay, what you see is you see a karyotype, and a karyotype just shows us all the chromosomes in every one of your cells. Okay, so if you see here, this is actually going to be a diploid cell. Okay, because you know this is a diploid cell because for every pair you have two chromosomes, one from mom, one from dad, or one from mom, one from dad. Okay, and if you continue all the way down and count them all, you have a total of 46. Okay, but what I really want to discuss is these little dashed lines within the chromosomes, okay? And those little dashed lines within the chromosomes are called genes. And genes are segments of DNA that code for specific traits, okay? So for example, if you look at this single chromosome right here, okay, what you see is all these different lines within this chromosome. And those lines are your genes, okay? And each one of these genes, okay, are going to code for a different trait. And you're probably like, well, what's a trait? A trait, for example, would be height or eye color, okay? So I'm just letting you know, okay, that each one of these is a single chromosome, and those dashed lines are going to be your genes. And those genes code for specific traits, all right? Okay, so beginning with the basics continued, okay? So now what we need to talk about are a couple more key vocabulary terms. Okay, so for example, what we're going to discuss is autosomal chromosomes, sex chromosomes, and homologous chromosomes. Autosomal chromosomes are every chromosome except for the sex chromosome. Okay, so autosomal chromosomes are pair 1, 2, 3, 4, all the way down to pair 22. Okay, and autosomal chromosomes relatively are about the same size and contain genes at the same location. Okay, now we're going to talk about sex chromosomes. Sex chromosomes, okay, are pair 23, which are right here. And you can always find the sex chromosomes in a karyotype because they are represented by the letters X and Y. And if you're thinking X and Y, male and female, you are correct. Okay, so here are your sex chromosomes. They're pair 23. And this chromosome pair right here actually is what determines your gender. They also contain genes on them. The X chromosome is going to have a little more genes. It's going to be bigger in size, okay, and it's going to contain a lot more genes than the Y chromosome. But essentially what you need to know here is that if you're a female, you have two X chromosomes, and if you are a male, you have an X and a Y chromosome. Okay, so this would be a male, this would be a female. Okay, also what we need to discuss before we get into talking about meiosis are homologous chromosomes. And homologous chromosomes are pairs of chromosomes that carry the same genes at the same location. So if you look here, these are all homologous chromosomes, okay? Homologous chromosomes, if you see here, they're all the same size and they all have genes, okay, at the same location coding for the same trait, okay? And primarily how we always recognize this is homologous chromosomes. You get one from mom and one from dad. Okay, and again here what you're looking at is you're looking at a pair of homologous chromosomes. They're similar in size and they have genes coding for the same trait at the same location. Okay, so these are homologous chromosomes right here. And again, in this karyotype, we see 22 pairs of homologous chromosomes. You get one from mom, one from dad. They're the same size and they have the same genes coding for the same trait at the same location. Okay, again, so here's a karyotype slide. Again, karyotypes are an ordered display of individuals' chromosomes. Okay, the staining them can reveal a visible band patterns which represent the genes, okay, which we talked about already. And the reason why we look at karyotypes is because it helps doctors identify any abnormalities such as mutations, okay? So again, if you look at the first image up here, okay, you can see all of the autosomal chromosomes 1 through 22, okay, and then you have your sex chromosomes XX and XY. Okay, this is a female because she has two X chromosomes here. And then you also see down here you have a male's genotype or a karyotype because you have an X chromosome here and a Y. Okay, so these again are your sex chromosomes. These above are your autosomal chromosomes. And notice that for every pair they're going to be are homologous chromosomes because they're relatively the same size and have the same bands at the same location coding for the same trait, which is super important. Alright, so on this slide we're looking at a karyotype for an individual who has the genetic disorder Down syndrome. Okay, so this is one way doctors can use a karyotype to help identify if an individual has a genetic disorder or not. Okay, so here is a karyotype, and essentially what this karyotype is showing us is this individual has the genetic disorder Down syndrome. And the reason why we know they have Down syndrome is because if you look at their 21st chromosomal pair, they have 3 chromosomes on this pair, okay? And essentially what we're seeing here is we're seeing an extra chromosome, and essentially what that is doing is it's giving that child the genetic disorder Down syndrome. So this is a way we can use karyotypes. So now what we're going to do is now we're going to get into talking about meiosis and the process in which meiosis occurs. So what is meiosis? Meiosis is the process in which gametes are produced, and remember gametes are our sperm cells and egg cells, which are haploid and contain 23 total chromosomes. Now how does this process work? Well, essentially the number of chromosomes is reduced from a diploid cell to a haploid cell. Okay, so essentially what we're doing in meiosis is we're going from a diploid cell with both mom and dad's genetic information, and we're producing 4 genetically different haploid sex cells, all containing 23 chromosomes each. And each cell is going to be slightly and genetically different from the others. That is the whole purpose of meiosis, to produce genetically different haploid sex cells. What is the summary process of meiosis? is where in meiosis we see two nuclear divisions, okay? And so in meiosis, we're going to actually have two cell divisions. We're going to have meiosis 1 and meiosis 2. And essentially what that allows us to do is it allows us to produce 4 genetically different sex cells. In males, it's going to be sperm cells. In females, it's going to be egg cells. And so what is the product? What are we producing at the end of meiosis? We're producing 4 genetically different sex cells. Males are sperm cells, females are egg cells. So on this slide, what we're looking at is we are looking at the stages of meiosis. And like I said on the last slide, in meiosis, there are going to be 2 different nuclear divisions, okay? So this first half over here is meiosis 1. This second half, okay, is going to be called meiosis 2. This process of meiosis still consists of IPMAT-C, okay, but it's going to be consisted of 2 nuclear divisions, okay? And the reason why is because we need to get to 4 genetically different haploid sex cells. So if you look here, you would still have interphase 1, okay, and interphase the DNA is in the chromatin form and so forth, and it's where the cell is growing. Then you have prophase 1, metaphase 1, anaphase 1, telophase 1, as well as cytokinesis. So at the end of meiosis 1, what we're producing is we're producing 2 haploid cells, but those haploid cells still have replicated chromosomes inside of them. Okay, so what we then need to do is we need to go through another stage of meiosis, which is meiosis 2, okay, to pull those replicated chromosomes apart so that we can produce 4 genetically different haploid sex cells, okay? So that is what we're going to get into next. So here is meiosis 1, okay, and essentially if you look here, it's still IPMAT-C. Okay, and so what we're going to do is we're just going to walk through the different stages of meiosis 1. Okay, so interphase 1, what we still see is we still see the chromatin is found inside the nucleus, so it's that spaghetti-like genetic material. The nuclear envelope is still present, okay, so we have a nice nuclear envelope there, and the DNA is replicating. Also, we have these centrioles that are replicating, okay, and the cell is growing. Next, we have prophase 1, and in prophase 1, this is where it gets a little different, okay, than mitosis, is that we begin to see homologous chromosomes aligning next to each other. Okay, so I'm going to just read off of my notes here. So in prophase 1, the chromatin is going to condense into chromosomes. So remember that in prophase, that is the first time where we see chromosomes present in the cell. And what those chromosomes are going to do once they condense is we're going to form homologous chromosomes. Okay, so here we have homologous chromosomes. We can say that blue is the male's chromosomes, red is the female's chromosomes. So we call those homologous chromosomes. So in prophase 1, these homologous chromosomes are going to go through this process called crossing over. Okay, and essentially what crossing over is it's this process right here. Okay, we have 2 homologous chromosomes that were just condensed in prophase 1, and what these homologous chromosomes are going to do is they're going to essentially wrap really tightly around one another, and as a result they're going to exchange part of their genetic material with one another. And the reason why we do this is because we need to increase genetic variability in each gamete produced at the end of meiosis. Okay, so as a result of this process of crossing over, we're increasing genetic variability. Okay, and essentially this process right here of crossing over is really what allows us to produce 4 genetically different sex cells. Okay, so again, this is crossing over and it occurs in prophase 1. So here in prophase 1, we have chromatin condenses into chromosomes. Homologous chromosomes are going to cross over, so they're going to swap genetic material with one another. And you can see that right here. The blue chromosome has a little red, and the red has a little blue. And then essentially, like mitosis, the nuclear envelope is going to break apart, and these chromosomes are going to begin to move within the cell. Okay, also you see these centrioles, okay, producing spindle fibers and moving to opposite ends. Metaphase 1. Metaphase chromosomes are going to line up in the middle. So in metaphase 1, homologous chromosomes are going to line up in the middle. Okay, and then also the spindle fibers are going to attach to the centromere of the chromosomes. Remember that the centromere is the center of the chromosomes. Okay, and you can see here that each chromosome, okay, has a little bit of each other's genetic material. Okay, next in anaphase 1, those homologous chromosomes are going to be pulled apart. Okay, so in anaphase 1, homologous chromosomes are pulled apart and moved to opposite ends of the cell. Okay, so we're essentially no longer having homologous chromosomes in the cell. They're being pulled apart. All right, so now we're going to get into meiosis 2. Okay, so at the end of meiosis 1, what we produce is we produce 2 cells that are haploid. Okay, and each cell contains 1 replicated chromosome for each pair. Okay, so you see 1 replicated chromosome here and 1 replicated chromosome here. Okay, and essentially we don't want replicated chromosomes in these cells, okay, within our sex cells. So what we have to do with these replicated chromosomes is we have to pull them apart. And this process right here of meiosis 2 is very similar to mitosis. Okay, so what I want you to also understand is that in meiosis 2, okay, the number of chromosomes in each cell is 23. However, they are just replicated. So in prophase 2 of meiosis 2, what's going to happen is, again, the chromatin is going to condense into chromosomes. The nuclear envelope is going to break apart and the centrioles are going to move apart. Meiosis 2, the chromosomes line up in the middle of the cell. The spindle fibers are going to attach to the centromere of each chromosome, okay, to align them down the middle. Anaphase, those replicated chromosomes are going to pull apart, okay, and are going to move to opposite ends of the cell. And as a result of this, what we produce is we produce 4 genetically different haploid sex cells. Each cell contains 23 chromosomes, and each one of these cells, which is very important to understand, are all genetically different. So here's a cool little animation to just show you the overall process. And here again is an overview of the different stages of meiosis. Okay, so we have interphase 1 up here. We have homologous chromosomes. Okay, next we have prophase 1. We have our homologous chromosomes lining up next to one another. And in prophase 1, remember that crossing over occurs, so they're going to swap parts of their chromosomes with one another. Metaphase 1, the homologous chromosomes are going to line down the middle of the cell. Anaphase 1, homologous chromosomes are pulled apart. At the end of meiosis 1, we're going to produce 2 haploid cells. Each haploid cell contains replicated chromosomes, and we don't want replicated chromosomes, okay? We want individual chromosomes, so what we have to do is go through another stage of meiosis, which is meiosis 2, and at the end of meiosis 2, okay, which is very similar to mitosis, we're going to produce 4 genetically different haploid sex cells, which you see down below. Okay, so here is oogenesis and spermatogenesis. Okay, so essentially what we're looking at here is we're looking at egg versus sperm cell formation. Okay, so oogenesis is how females produce their egg cells. Now, the production of females' eggs, okay, is slightly different than males. Okay, males are constantly producing sperm cells, okay, over time. Okay, so males produce millions and millions of sperm cells throughout their lives. Females, excuse me, are actually born with their egg cells. Those egg cells, however, as they mature, okay, are going to develop into these fertile egg cells, which can then fertilize with a male sperm cell, okay, to form and to have a child. Okay, but what I really want you to understand and see from this video or this diagram is that each one of these sperm cells down below that are produced at the end of meiosis, okay, are all going to be genetically different. They're all going to have a different genetic code, and that's so important because if we produce sperm that are identical, that ultimately could produce identical children, which is not good. Okay, that increases the likelihood of us being susceptible to the same diseases, and that is not the point of sex and meiosis. The point of sex and meiosis is to produce genetically different offspring so that we produce genetically different individuals. That is all I have. If you have any questions, please email me. Thank you very much.